This is Unit 7, Part 4. Now, previously we have spoken about blood flow through the entire circulation. And in fact, the flow through the systemic circulation is equivalent to the cardiac output. And now we'll talk about flow through particular different organs and how that can be locally controlled within, so that the flow into the particular organ can be varied. And then we'll talk about flow through blood vessels, individual blood vessels, how that can be altered somewhat. All right, the first tissue that or organ systems we'll look at are skeletal muscles. Now, in resting muscle, blood flow into the tissue, and skeletal muscle does form a large part. All of the different skeletal muscles do together form a large part of the body mass. Blood flow into that resting muscle tissue is regulated by myogenic mechanisms and general neural mechanisms that respond to the O2 and CO2 levels in the tissue. As muscle becomes active, as it becomes more metabolically active, and in the case of muscle that's due to contraction, then there's a very quick response of what's called hyperemia, which is an increase in the flow of blood into that tissue. So as soon as you start using your muscles, the blood flow increases. You have hyperemia, increased blood flow. And the increase in the blood flow is directly proportional to the increase in metabolic activity of the muscle. You start walking, you have more blood flow in. You start running, you have even more flow in. This is called also called active hyperemia or exercise hyperemia. So the body responds very nicely, quickly to skeletal muscle being used, needing more oxygen, because once the skeletal muscle is active, it's going to need to generate a lot of energy, and therefore will need a lot of oxygen. The starting up of muscle activity involves often triggering of the sympathetic nervous system, the sympathetic division of the autonomic nervous system, right? I'm sorry, that's wrong. That was wrong what I just said. Increase in muscle activity, it's voluntary tissue, is associated with an increased output of the nervous system that controls voluntary tissues. And so you get an increase in release of adrenaline or epinephrine, and that can bind to receptors in the smooth muscle around arterioles. So, and that causes a vasodilation. So you get a relaxation of that muscle, vasodilation, so the blood flow into that muscle increases. And so the idea here is to, going from rest to activity in skeletal muscle, you have an increased flow. The arterioles, in fact, have neurotransmitter receptors. They respond to these chemical signals sent out by the nervous system. And those receptors can respond to either acetylcholine, those are called cholinergic receptors, or to adrenaline, epinephrine, those are called adrenergic receptors. And there are different types, alpha and beta, and there are subtypes of alpha, etc. These alpha, beta, adrenergic receptors can bind adrenaline or epinephrine. Those two are the same, right? And respond in that way. If you have a low level of epinephrine release, it binds to beta-2 receptors. That causes a vasodilation, so you get increased blood flow into the muscle. As the increase activity is triggering by increased neurotransmitter release and increased responses. Blood flow can increase as much as 10 times the normal amount at rest. And so you get a massive increase, tenfold increase in blood flow because of the physical activity requiring that increase in delivery of oxygen, and therefore you have vasodilation and increased blood flow. However, that increase in flow in can result in or be associated with higher and higher levels of adrenaline or epinephrine release. And at very high levels of epinephrine, you start getting the binding of the epinephrine to alpha adrenergic receptors. Okay, now binding to the alpha adrenergic receptors results in local vasoconstriction. And this is a protective response to prevent the muscle's activity and demand on oxygen from exceeding the cardiovascular ability to supply it. So you don't want it to exceed the ability of the heart to pump out blood to provide adequate cardiac output of blood, like going beyond the heart's ability. So at lower levels, you have adrenaline release associated with that, because as soon as you start acting, you're releasing more and more epinephrine or adrenaline, and that will cause vasodilation. But if you're at a very intense level of activity, a high level of activity, you start getting binding to these alpha receptors, and you start getting some vasoconstriction. The blood vessels start narrowing even. Actually, and that kind of cuts off the supply of blood, not completely, but it reduces the supply of blood to the muscles, and they fatigue and they stop, because you want control regulation at that level. You don't want to exceed your heart's ability to provide blood or pump it out. Okay, in terms of that was skeletal muscle. In terms of the brain, blood flow to the brain needs to be constant. Neurons have a very high demand and requirement for oxygen all the time. They are extremely intolerant of ischemia, inadequate blood supply. They always need oxygen. They always need to be provided with oxygen. You don't last long if the oxygen supply to your brain is reduced below adequate levels. It's extremely sensitive. In fact, it responds to metabolism overall activity. When a tissue is more active, you will get an increase in hydrogen ion release. So the hydrogen ion concentration goes up, which means that the pH will drop. And a drop in pH is associated, in fact, with marked vasodilation of the arterioles feeding into the different areas of the brain, because that'll supply more oxygen. You get a drop in pH, in other words, an increase in hydrogen ion concentration, when you have a higher level of CO2. Remember, you get a higher level of CO2 when a tissue is more active metabolically. So the increase in metabolic activity, increased activity in the brain and other organs will generate more CO2, triggering a drop in pH, and that will immediately trigger a response of vasodilation, so you get more blood flowing in. This kind of regulatory response to the metabolism is very important. There's also myogenic controls that protect the brain from any damaging shifts in blood pressure. A decrease in mean arterial pressure, overall mean arterial pressure, will trigger the blood vessels that supply the brain to dilate. They'll vasodilate to ensure adequate perfusion. And if the mean arterial pressure is too high, you'll get those blood vessels constricting, undergoing vasoconstriction, to ensure that you don't get too much high pressure in the brain, because that can also cause injury or damage by causing edema or swelling and loss of function. Remember, the brain is inside a hard case, so it can't swell outwards. So decrease mean arterial pressure, get vasodilation. Increase mean arterial pressure, you get vasoconstriction in the brain right away in response. The brain can, in fact, regulate its own blood in certain circumstances, such as ischemia and blood flow in certain circumstances, such as ischemia caused by a tumor. You know, as I said, the brain is inside a hard case, and if you have a tumor growing in there, then it will cause pressure that can't be relieved by the brain swelling outwards because of the hard case. And so what happens is there's increased pressure on the tissues. So in that response, at least, you will get some ischemia in an area of the brain, and actually that can trigger symptoms. This is not really, this is such a, brain tumors do not occur, is neither normal nor occurring that frequently that we should talk about it. The main thing to realize is that the brain is particularly vulnerable to extremes in overall systemic blood pressure. When there are changes that are extreme, one of the very first things is you faint. So if your mean arterial pressure drops below 60, boom, you have fainting, which is properly called syncope. If mean arterial pressure goes above 160, that can start resulting in movement of blood fluids out of the circulation into the brain tissue itself, and you get swelling or edema, cerebral edema, and that can cause problems. Okay, in the skin, response is a little different in terms of blood flow through the skin. The blood typically supplies nutrients to the cells just like everywhere else, and will fulfill the oxygen requirements. But it also is very important, blood flow in the skin is very important in helping maintain body temperature. That's one, these kind of things are what's happening pretty much everywhere in the body. But in addition, the skin, which is the body's largest organ, is very important in maintaining body temperature. The skin can also act as a reservoir of blood, because it can actually have enough vasodilation or vasoconstriction to shift more blood into the rest of the circulation, or less blood into the rest of the circulation. Blood flow in the skin is also not just through, the usual arteries, arterioles, capillaries, veins, and venules and veins, but also into these large expanded areas called plexuses, or networks, venous plexuses just below the skin surface. And the flow can vary tremendously depending on body temperature. It can range, here you see the range can be 50-fold, from 50 milliliters per minute to 50 times that, which is 2,500 milliliters per minute. This kind of variation in flow into the skin just below the surface, into these areas of the just underneath the surface of the skin, is controlled by the sympathetic division of the autonomic nervous system. These are reflexes that are typically initiated by temperature receptors, which send signals back to the central nervous system. Then the central nervous system sends out messages through the sympathetic division of the autonomic nervous system. If you are hot, have a fever, or you're vigorously exercising, you've been exposed to a high temperature outside, then you get a reduction in stimulation of the muscle in the blood vessels, so vasomotor stimulation is reduced. You get vasodilation and more blood flow into the skin, and heat gets radiated away from the skin. By shunting of that blood to the surface, you can radiate it away, and you also sweat, which also helps with losing heat because you evaporate the water in the sweat, and it pulls heat away from you. Also, sweat can directly cause vasodilation by the bradykinin in it. They stimulate the release of nitric oxide. Nitric oxide can cause further vasodilation. So in response to that blood flow more to the surface of your body, you're losing heat, and body temperature drops, and then the blood is shunted down back to a normal situation where less blood is at the surface, and it's flowing throughout the rest of the body at normal levels. That's high temperature responses. Let's move to the lungs. Oh, and by the way, I should mention, of course, that if you're cold, then in a cold temperature, then you shunt the blood away from the surface so you don't lose extra heat. Blood flow in the lungs is unusual because, first of all, the overall pathway from the heart to the lungs and back to the heart, the pulmonary circuit, is fairly short. The arteries and arterioles are more like veins and venules. They're bringing a lower pressure blood up to the lungs to get, and they're carrying blood that's less than fully saturated with oxygen to pick up oxygen and drop off CO2. So they do have a higher pressure than arterial pressure, but it is nowhere as high as the systemic system, the arterial side of the systemic system, systemic circulation, which is at 120/80. And the pulmonary circuit, the arterial pressure is only 24/8. But that blood is being pumped up from the right ventricle up through the pulmonary arteries to the heart, to the lungs, to get oxygenated. You need to deliver that blood to areas of the lung that are being ventilated well. The blood needs to get to an area that has good ventilation, good flow of air in it. There's a mechanism, in fact, that will deliver more blood to the best ventilated areas and less blood to those areas of the lungs that may not be as well ventilated. You don't want to send the blood off to an area that's not getting good airflow. This is an autoregulatory mechanism, and it's exactly the opposite in most other tissues. In most other tissues, when you have inadequate amounts of oxygen, you get more blood flow into them, more arterial blood flow. If you have somewhat higher oxygen in the tissue, then it doesn't need as much blood flow, arterial blood flow in. But in the lungs, it's completely the opposite. Areas, as I said before, with low oxygen levels in the lungs, those will have the fact that there's a lower oxygen level in that particular area of the lung will trigger vasoconstriction of the blood vessels going to that area. You don't want to send the blood there because it's not going to be able to pick up as much oxygen. It'll go instead to the higher, well ventilated areas, so areas of the lungs that have a lot of oxygen. That triggers vasodilation and more blood flows to that. In this way, it guarantees proper oxygen loading of the blood in the lungs, because the blood is going to flow more to areas that get more flow of air. In other words, better oxygen. Okay, let's go to the heart. In the heart, small blood vessels on the arterial side, the coronary circulation there, most of they're influenced by aortic blood pressure. Pumping activity of the ventricles is important to consider also, because you have to realize that these small blood vessels that supply the coronary circulation, the vessels that stretch out over the surface of the heart and then dive down into the heart muscle itself, when the heart contracts, they're squeezed quite hard. So in fact, during ventricular systole, contraction of the ventricles, the coronary blood vessels are quite compressed, and in fact myocardial blood flow ceases. It actually stops temporarily because of that contraction, squeezing, hard contraction of the ventricles. In that case, there you do have myoglobin, the red oxygen carrying molecule, red pigment colored molecule in the heart muscle itself that can supply oxygen. There is enough blood flow in the coronary circulation during diastole to supply sufficient oxygen and nutrients. So when the ventricles contract during diastole, blood flows easily and oxygen nutrients are carried to the muscle. When you're in ventricular systole, it doesn't flow. The vessels are compressed. Under resting conditions, the blood flow through the heart can be controlled locally by myogenic mechanisms that are under myogenic control. When that's resting, if strenuous exercise starts, so that the heart starts to beat faster and harder, then the coronary vessels will dilate due to increased carbon dioxide in the tissues. The PCO2 goes up because the heart muscle is using is generating more energy, and therefore generating and releasing more carbon dioxide, CO2, from the breakdown of carbohydrates, like glucose. Blood flow can increase 3 to 4 times into the coronary circulation because of that dilation, and it needs to increase to supply that extra required oxygen for energy production. Normally, blood flow overall remains constant, even though there can be wide variations in coronary perfusion pressure. All right, now we're going to move on to a different topic, and that's local capillary function. And the main function, of course, of the capillary is to exchange respiratory gases and nutrients with the tissue, and also pick up metabolic waste. So now we'll just be focusing on capillaries and what the pressures functions are that affect flow or diffusion of these substances across the capillary wall, from blood to tissue or from tissue to blood. Now, there is diffusion of each of these components between the blood and the interstitial fluid along their concentration gradients. Concentration of oxygen, the PO2 of oxygen in the capillary blood as it enters into the capillary network, is higher than in the tissues, so it diffuses out into the tissues. CO2 level, PCO2 in other words, is higher in the tissues than in the blood, so it diffuses into the blood. Nutrients are diffusing from the blood into the tissues. Metabolic wastes are diffusing from the tissues into the blood. Okay. Now, water-soluble solutes can pass through in the clefts that exist between endothelial cells that line or pave the wall of the capillaries, and there are also fenestrations, openings that can allow water-soluble solute molecules through. Lipid soluble molecules, on the other hand, can diffuse directly through the membranes, the cell membranes of endothelial cells, because there's nothing stopping them. They can actually move right through that phospholipid bilayer. And here you see the capillary, what it looks like. This is happens to be a more fenestrated open capillary. There are also, there are different types of capillaries, but all allow for some degree of, certainly a fairly efficient degree of exchange between the blood and the tissue and vice versa. What you have, of course, as I said, are intercellular clefts here and some fenestrations, more depending on whether it's a tight or closed capillary or a fenestrated capillary. The fluid movement, really a lot of it depends on hydrostatic pressure and colloid osmotic pressure. Capillary hydrostatic pressure is generated by the blood pressure, but you also have some pressure, fluid pressure within the tissues. If it's higher in the capillary, if the hydrostatic pressure is higher in the capillary, then that's pushing outwards. That's a force pushing outwards, as compared to the force fluid capillary hydrostatic pressure or interstitial hydrostatic pressure pushing inwards into the capillary. So it's a question of, forces one way or the other. There is also what's called osmotic pressure, and that's the ability to pull fluids, water, by osmosis. Diffusion of water is called osmosis, and there are molecules within the blood plasma that cannot, they're too large to escape the blood plasma, and therefore fluid does get pushed out, but these solutes that are too large to move out do have the ability to pull fluid in, to pull water in by osmosis. That's called colloid osmotic pressure. So the capillary, inside the capillary, there is a colloid osmotic pressure, mostly generated by the molecule albumin, a protein molecule that's fairly large and makes up about 50% of the plasma proteins. So the hydrostatic pressure of the blood is pushing outwards against the capillary walls, tending to force fluid out, not cells, but fluid out through the capillary walls. And it's always greater at the arterial end, the beginning end of the capillary bed, than at the venular end. The capillary colloid osmotic pressure, sometimes called oncotic. You may see this term in some places, oncotic. Oncotic pressure means the same as colloid osmotic pressure, or the oncotic pressure. The oncotic pressure is created by, as I said, non-diffusible plasma proteins that can't get out, but are able to pull water in through, by osmosis, pull water towards them. So what you see is this kind of effect. Here we have, this is the arterial end where blood is entering into the capillary from the arterioles. It's going this way, that's the blood flow direction, and then out this way at the venular end or venous end of the capillary. So blood flows in, and the hydrostatic pressure at that end is 35, and it pushes fluid out. And at the venous end, it is 17. So you have 35 here pushing out, and then 17 pushing out. It's much lower because quite a bit of the fluid has gotten pushed out into the tissues, in the space between the tissues. So it's going into the interstitial space. It becomes interstitial fluid. And that interstitial fluid that gets pushed out is the same as plasma minus the proteins, okay? So it gets pushed out. And then you have the interstitial fluid hydrostatic pressure. You have to consider that. At this end, it is, in fact, 0. And here it is, we're still talking 0, okay? So the net hydrostatic pressure at this end, at the arterial end of the capillary, is 35, and the net hydrostatic pressure at towards the end of the capillary bed, towards the venous end of the capillary bed, is 17. Okay. Now, in terms of the colloid osmotic pressure, it is 26 here. The interstitial colloid osmotic pressure is 1, and again, it's here. It may increase somewhat, actually, at this end. In some cases, you'll see examples where they're showing some increase, because after all, if more fluid flows out, then there's a higher protein amount here, a concentration of protein. But generally, it pulls fluid in, and the net osmotic pressure colloid osmotic pressure is 25 inwards. Hydrostatic pressure net is 35 out, and therefore you have a net filtration pressure at this end of 10. And at this end, the hydrostatic pressure has been reduced because of the loss of fluid, so pressure is somewhat lower, and the osmotic pressure is still 25, so it's 17 minus 26. You have to consider this, 1. So you have a minus net filtration pressure, and that means that you have an inward pressure. Here it's an outward pressure, and it's slightly bigger than the inward pulling pressure, which means that some fluid stays in the interstitium. So as blood flows into this capillary, fluid is getting pushed out, and some of it doesn't get pulled back in. Most of it gets pulled back in further down the capillary towards the venous end, because of this inward net filtration pressure. First, it's pushed out by the net filtration pressure pushing outward, then it's pulled back in, but not all of it gets pulled back in. Some of it stays. You may see in other textbooks, other figures, other slides, other sources, slightly different numbers, but in the end, the general message is that the net filtration pressure is higher at the arterial end. Fluid gets pushed out, then it gets pulled back in by the action of the colloid osmotic pressure of the albumin, mostly is the key protein. So it's getting pulled back in, but not all of it. In the end, you have some fluid left out because the net outward pressure is greater than the net inward. This kind of looking at the net filtration pressure, it considers all of the forces acting on the fluids in the capillary bed. The capillary hydrostatic pressure, which is an outward pressure, the interstitial fluid hydrostatic pressure, which is an inward pressure, and is always raised if there's edema, if the tissue is swelled up, there's more. The colloid osmotic pressure, which is inward pressure, then the interstitial fluid osmotic pressure, which is can pull some fluid out. But the end result is you can take all of these into consideration to determine the net filtration pressure, and the equation is this. This tells you the overall net hydrostatic pressure, and this tells you the net overall colloid osmotic pressure, and the end result is a slight advantage to outward pressure versus in. So there is a overall net outward pressure. At the arterial end, the hydrostatic forces dominate, and at the venous end, the osmotic forces dominate. So more fluid flows in, but not as much flows in as flowed out. Slightly more flows out than flows back in. However, that excess fluid does not accumulate and you don't swell up. The reason is because you have a drainage system. You have the lymphatic system that can drain that interstitial fluid and return it through that network of lymphatic vessels back into the blood circulation. It'll enter into the blood circulation because the lymphatic system joins up and drains into the right subclavian vein and the left subclavian vein. The end result of this, I should point out, is that you have kind of a gentle wash of fluids leaving the capillaries, which are everywhere in the body. So there's a gentle sort of flush of fluids leaving the capillaries, washing through the interstitial space into the lymphatic vessels. And the lymphatic vessels lead along, and every once in a while, quite a few of these, you have these lymph nodes where the fluid can interact with cells of the immune system. All the different players, all the different cells of the immune system are there. And if there's any foreign molecule that got flushed into that, then it can interact with immune cells and you can develop an immune response in that way. We'll talk about that more in Unit 13. But the fluid flows through, and every once in a while there are these filters of lymph nodes acting on the fluid and interacting with foreign molecules if there are any, and the fluid goes along and then it joins back up with the blood circulation, so it all flows back into the blood in the upper chest at the right subclavian vein and the left subclavian vein. And so you have this nice gentle wash through all of your interstitial fluid. Sometimes, we're back onto the blood in general, sometimes there are conditions quite serious where you can get circulatory shock, where there is inadequate filling of the blood vessels. And if you have inadequate filling, then you'll get a low blood pressure and the blood can't circulate properly. You get inadequate blood flow to meet the demands of the tissues. They will not be getting enough blood supply. They will not get enough oxygen to function. Things will go bad fast, really fast. This can be due to blood loss, or it could be due to widespread vasodilation and increased vascular permeability, so that more fluids are going out of the circulation and into the tissues. Circulatory shock is very serious. There are generally there are three types. There's hypovolemic shock from large-scale blood loss. There's vascular shock resulting from, and I mentioned this also just now, from a large-scale widespread vasodilation. And then there's cardiogenic shock, which is because the heart can't sustain adequate circulation. The heart muscle tissue is injured or damaged, or some problem going on. So hypovolemic, vascular, and cardiogenic are three types of shock that can generate circulatory shock. This is a chart that shows you the responses of the body, the regulatory responses of the body in the case of hypovolemic shock due to a large loss of blood. I'd say it would be worse than just some little glass cut like this on somebody's foot. In order to, I mean, they're not going to show like somebody with their leg cut open at the femoral vein, at the femoral artery, and spurting blood. They showed this picture, but it takes more than just that to get a hypovolemic shock due to acute bleeding, larger volumes. Anyhow, you can go through this, and really we've talked about a lot of this and a lot of the responses, and I think it's worth thinking about each of the different steps. Just follow the trail and see what happens and see if it makes sense to you. Okay, these next few slides from here to the end are either a review of what we've done before, what we've discussed before in terms of the pulmonary circuit and the systemic circuit, and the some detail on the pulmonary circuit, which we've also gone through before, and then just an overview. And then there's quite a few diagrams of detailed blood vessels in different parts of the body, much more than I would ever expect you to know, but this way you have the diagrams, and if you ever need them or want to look at them, you can have them and keep them. I'll go through in the next few slides certain particular structures which we've talked about before, just a few. The vascular system, of course, has two distinct circulations. We've done this before. The pulmonary circulation, a shorter loop from the heart to lungs and back, and the circulatories with a lower blood pressure than the systemic circulation. It's the systemic circulation is a larger route that pervades or perfuses all parts of the body, returning to the heart. Differences are major differences between arteries, the arterial side, and the venous side is that on the arterial side, it's blood's pumped into a single systemic artery called the aorta, and that distributes that blood throughout the systemic circulation. On the venous side, the blood returns via the coronary, via the superior vena cava and the inferior vena cava, and also, of course, the coronary sinus. Arteries tend to be deeper and better protected in terms of being surrounded by more tissue because they're deeper into the tissues. Veins are more superficial. If you get a cut, odds are you'll cut a vein rather than an artery. That's why it oozes rather than spurts. The pathways for the arteries are fairly clear and well defined, whereas with veins you get convergence and divergence and interconnections. There's many possible routes of the blood to, there's only one route typically for blood to flow to a particular tissue along the arterial side, but along the venous side there are alternate routes of drainage. So supply on the arterial side is very clear cut, and drainage is somewhat less defined. You have these sinus areas. There's also the hepatic portal circulation to consider. Doesn't the blood draining out of the tissues in the abdomen first go into the liver through the hepatic portal vein and get processed and then go back to the heart? There are some unique developmental aspects in terms of the circulatory system, many of which we've spoken about, so I won't go into in the lab, so I won't go into great detail on them right now, but we do cover them also in the lab in a fair amount of detail, and I'll run through it quickly. First of all, in terms of development in the fetus, endothelial cells come from a particular primitive type of cell that happens to be called mesodermal cells. I don't think that's that key for you to know. You get these at the very, very early stage, the tiny fetus, very early developing fetus. You get these blood islands and a little area that starts to pump, which will develop into the heart, and that's by the 4th week of development. You have this tiny thing that's going to be the heart pumping through these areas that will be blood vessels. You do, of course, get development of the umbilical vein and arteries that circulate blood back and forth from the placenta, where the maternal circulation and the fetal circulation are close to each other, so you can get good exchange by diffusion of oxygen, carbon dioxide, nutrients, and metabolic waste products. Fetal shunts that are present only in the fetus are important to understand. Realize there is a shunting around the lungs. There is blood sent to the lungs from the heart, but not that much because the lungs aren't breathing any air. So you do have the foramen ovale, which is a hole formed in the wall between the right atrium and the left atrium. There is also the ductus arteriosus, which is a shunt bypassing the blood gets pushed out of the heart, pumped out of the heart up the pulmonary trunk, but most of it doesn't go to the lungs. It goes over through a shunt into the aorta. And that shunt, that structure is called the ductus arteriosus. There is also a bypass of the liver. The main vessel directing blood towards the liver has a shunt, so it doesn't go into the liver, but rather goes into the inferior vena cava and back to the heart, because the liver doesn't have to process that blood. And that shunt is called the ductus venosus. Blood vessels typically in youth, young people, are nice, trouble-free things. The formation of blood vessels occurs in order to support the growth of the body and wound healing and rebuilding muscle vessels does occur somewhat, only to any significant extent in females during the menstrual cycle. But with aging, you get weakness in the walls of veins so that they widen out unnaturally. You get varicose veins. You get some deposits and calcium streaking in the arterial side, which are characteristic of a process called atherosclerosis. And overall, you typically get an increase in blood pressure. Here's the pulmonary circuit, where the blood goes up through the pulmonary trunk, through the pulmonary, out of the right ventricle, up the pulmonary trunk to the right and left pulmonary arteries in the right and left lungs. And then the gas exchange occurs in the right and the left lung, and then goes back to the left atrium, and then down to the left ventricle and out through the aorta to the systemic circulation. And here you see a diagram showing that, and you're familiar with that. And you should, of course, understand that there are a couple of blood supplies to the to the lungs, not just from the pulmonary trunk and the pulmonary arteries for blood to be oxygenated and then return to the heart, but also there is a branch from the aorta that will supply the bronchial circulation to the lungs, because there are some parts of the lung tissue that aren't in close enough contact with air. They need their own arterial supply. But blood goes up and gets, most of it is in this pulmonary circulation circuit, goes up through pulmonary arteries, then gets oxygenated and goes to pulmonary veins and returns to the heart through these pulmonary veins on the left and on the right, and returns to the left atrium, the left ventricle, and gets pumped out the aorta to the rest of the body. And here you see again just a diagram, detailing, in this case, not the pulmonary circulation, but the pulmonary circulation is shown in gray, but this is the systemic circulation, showing the venous side and the arterial side in blue and red, respectively, and showing that it's an artist's diagram. There are capillary beds everywhere in the entire body. You need gas exchange everywhere to occur. This is a diagram showing major arteries, the aorta. Of course, we have the three major branches off the aortic arch of the brachiocephalic trunk, which will then divide into the right common carotid. I don't know if that's, they're showing them together. The right common carotid and the left, and then also the right subclavian. Is it labeled? No. Anyhow, I'm going to draw it for you. Okay, there's the aorta going up. This is not a very good drawing, but here is the aorta going up like that. And the first major branch off of it is the brachiocephalic trunk, which then divides into the right common carotid artery and the right subclavian. And then the next branch going upwards is the left common carotid, and the third major branch off of that is the sorry, this is the right common carotid artery, this is the left common carotid artery, and this is the left subclavian. Okay, there's the right subclavian right there. And that's not that clear here. Let's see if we can see a better one. Here's this is a side view, and it's the left side, so it's the left common carotid artery. There's the left subclavian artery. There's the brachiocephalic. Is this the? No, this is the right side. This is the right side of the face. So that's the right common carotid artery, right subclavian artery, which branch off of the brachiocephalic trunk. It's not very well drawn because you can't see the the other ones going off. Okay. I'm not going to get into any of these, but you have the diagram. You can, if you ever need it or want it, you have it. This one's, I guess, a little bit better, showing the brachiocephalic trunk on the right side than the right common carotid artery and the right subclavian artery, and that's off of the brachiocephalic trunk on the right side. This is the right side. This is the, actually, let's not label it that way. Let's label it like this. This is the right side, and this is the left side. And we're looking at brachiocephalic trunk splitting into the right subclavian and the right common carotid. And then you have the left common carotid artery, and you have the left subclavian artery. It's important to say left or right, and artery or vein. Okay. You should know the brachial artery, of course, and the radial artery. The brachial artery is used for determining blood pressure. It's in the upper arm there, and to draw blood, you'd use the vein, but the brachial artery is used for determining blood pressure. That's where you put the cuff. And the radial artery is used for determining pulse at the wrist. Here you see arteries in the abdomen. I think I can just keep going here. Abdominal. These are retroperitoneal organs. This is showing lots of arteries in the mesentery, supplying arterial blood to the large intestine. They're showing mostly large intestine here. They've stripped away the small intestine. There's the very end of the small intestine, right? There's the appendix. All of those are on the right side of your body. There's the femoral artery in the groin. That's you can take a pulse off, and sometimes they will take arterial blood samples from there. Okay, in terms of the venous circulation, I have mentioned many times the hepatic portal vein, which drains all the blood from the abdominal organs into the liver, and then that gets processed in the liver and leaves the liver through the hepatic veins that lead to the inferior vena cava and then there's also the superior vena cava draining the upper body. You have these diagrams. They may be useful for you. I wanted you to have them. Some of these are used for drawing blood. I'm assuming it's the median cubital vein that often is used. And that's about it. There's a hepatic portal vein leading into the liver. Here, all of this is liver, right? All that is the liver. And there's the hepatic portal vein leading into the liver, draining all the organs. So the liver will process the nutrients and take care of some toxins and kill off any bacteria that might have snuck across the wall of particularly the large intestine. There are a huge number of bacteria in the fecal material inside the large intestine that have to be taken care of. And we get the last slide. So that's the end of Unit 7 on the vascular system, the vascular part of the cardiovascular system.